Today, we're going to talk about a scientific revolution that was sparked by a man who thought the whole idea was, well, complete nonsense. His name was Thomas Hunt Morgan, and his incredible journey from being a total skeptic to a biology pioneer would completely rewrite our understanding of life itself.
I mean, it's a story that really makes you wonder. How does a top scientist go from calling a groundbreaking theory an absurdity to winning a Nobel Prize for proving it? Well, the answer involves a ton of patience, a really sharp eye, and, believe it or not, millions of tiny fruit flies. So to really get a sense of how huge Morgan's breakthrough was, we've got to go back in time to the turn of the 20th century. Back then, the world of biology was kind of stumped by this grand genetic mystery. All the pieces of the puzzle were starting to come together. You had Gregor Mendel, who'd figured out that traits were passed down by these factors, what we now call genes. Then, scientists actually saw chromosomes inside cells, and they seemed to move in patterns that perfectly matched Mendel's laws. So, by 1902, it looked like they had it figured out. The chromosomal theory of inheritance. The idea was simple. Chromosomes carry the genes. It was neat, it was elegant, but there was one massive, glaring problem nobody could figure out. And here was the big issue. Think about it. Any organism has thousands of different traits. Eye color, height, wing shape, all of it. But when scientists looked under a microscope, they could only see a pretty small number of chromosome pairs. So how could just a handful of chromosomes carry the instructions for thousands upon thousands of traits? The math just didn't work, and for a lot of people, the whole theory just fell apart right there. And this is where our main character, Thomas Hunt Morgan, enters the story. But he wasn't coming in to save the day for the chromosomal theory. Oh no, he was one of its loudest and most respected critics. Morgan, you see, did not mince words. He thought the whole idea that genes were physically sitting on chromosomes was, and I quote, an absurdity. He was an experimentalist, hands-on guy, and a theory just didn't square with the facts he could see. So he decided to figure out how inheritance really worked on his own terms. He just needed the right test subject. And he found it in the humble fruit fly. Seriously, they were perfect for the job. You could raise them by the million in old milk bottles, they produced a whole new generation every 2 weeks, and they had a really simple genetic code, only 4 pairs of chromosomes. His lab at Columbia University quickly became known as the fly bro. So for 2 long years, Morgan and his students were just surrounded by a sea of red eyes. They were breeding millions and millions of flies, generation after generation, just staring at them, looking for one single random mutation, any little thing that was different from the rest. It was tedious, painstaking work. And then, after all that waiting, the revolution began. And it didn't start with a huge explosion, but with one tiny little white-eyed fly. In May of 1910, it happened. In a bottle filled with countless red-eyed flies, there was one single male with pure white eyes. This was it. This was the mutation he'd been hoping for. Morgan snatched that fly up and immediately started breeding it, kicking off a series of experiments that would change biology forever. But as he started breeding this unique little fly, a really strange pattern popped up. This white-eyed trait, it wasn't following the simple rules that Mendel had laid out. Instead, it was showing up almost exclusively in the male flies. Now, why on earth would eye color be tied to sex? This was a completely new puzzle. And then Morgan, the great skeptic himself, had this incredible flash of insight. He realized the only way this weird inheritance pattern could possibly make sense was if the gene for eye color was physically located on the exact chromosome that determines sex, the X chromosome. This was the moment. For the very first time, a specific trait was linked to a specific chromosome. He called this discovery sex linkage, and with that one little white-eyed fly, he found the first concrete proof for the very theory he once called an absurdity. In trying to disprove it, Morgan had accidentally become its biggest champion. But proving genes were on chromosomes wasn't the end of the story, not by a long shot. It was just the beginning. I mean, if a single chromosome carried a bunch of genes, how were they arranged? And what was keeping them together? See, Morgan's team noticed that some traits seemed to be inherited together way more often than you'd expect by chance. This led them to the idea of genetic linkage. Basically, genes that are physically close to each other on a chromosome kind of get stuck together and are usually passed on as a package deal. It was a great explanation for how one chromosome could carry info from many traits. But here's the thing. they weren't always inherited together. Sometimes these linked genes would mysteriously separate. Morgan figured this was because of something he called crossing over, a process where pairs of chromosomes actually swap pieces with each other. It's like a genetic shuffling of the deck that could break up those linked genes. Now, this is where one of Morgan's undergraduate students, a guy named Alfred Sturtevant, had a truly stunning insight. He realized that this crossing over wasn't just random, it was a clue. He had this idea that the frequency, how often these swaps happened, could tell you something profound about how the genes were physically laid out. Sturtevant's logic was so simple, yet so brilliant. Imagine genes are like towns along a highway. A crossover event is like a random road closure popping up somewhere on that highway. Now, if two towns are really far apart, there's a good chance the closure will happen somewhere between them. But if they're right next to each other, it's really unlikely. So, by just counting how often two genes got separated by a road closure, he could actually map their relative distances from each other. This single idea from Sturtevant, born from all that work in Morgan's fly room, it just blew the doors open on a whole new way of looking at the genome, and it cemented Morgan's place in history forever. And in 1913, Sturtevant published it, the world's very first genetic map. It was just a simple little diagram showing the linear order of 6 genes on the fruit fly's X chromosome. But this was absolutely revolutionary. The gene was no longer just an abstract idea, now it had a physical address. This work was so foundational that the unit we use to measure genetic distance is named in Morgan's honor, the centimorgan. 1 centimorgan is basically a 1% chance that 2 genes will be separated by recombination. It's a standard that geneticists still use to this very day. And, of course, the ultimate recognition came in 1933. The guy who was once the biggest skeptic, Thomas Hunt Morgan, received the Nobel Prize for his discoveries about the role of the chromosome in heredity. What a complete 180. His legacy is just immense. He and his team didn't just prove the chromosomal theory was right, they gave us the concepts of sex linkage, genetic linkage, and crossing over. They basically invented the entire field of genetic mapping and turned the humble fruit fly into a cornerstone of biological research. Morgan really did lay the foundation for the next 100 years of genetics. Think about it. We went from mapping 6 little fly genes to sequencing the entire human genome. The progress has just been breathtaking. And it all started in that little fly room, driven by a skeptic who trusted evidence more than anything else. It really leaves us with a big question today, doesn't it? In our own time, what absurd ideas are just one white-eyed fly away from changing the world? What's our next frontier?